apologise to those who had questions to ask, which there wasn't time to fit in, but we've got a whole weekend, so please grab me. There are going to be a couple of good long discussion sessions over the course of the weekend, so it should be possible to have your say. So we're really extremely lucky to have Joel with us. I think probably travelled further than anyone else, coming all the way from Sydney. So Joel is one of the world's leading imagery researchers. Joel, you got in touch with me originally, though I knew about you because Aphantasia had swum past him and he was interested. Joel has developed techniques which I'm sure he'll talk about for measuring imagery in objective ways, which is, of course, challenging. And he worked on that in Sydney for a number of years with a background actually in the arts. I was being reminded last night that Joel was at art college for a while before moving into psychology. It's a background that equips him very well for studying imagery. And recently he's taken something of a new departure and is now working in collaboration with startups. So he's director of the Future Minds Lab and head of innovation and enterprise at the School of Psychology, University of New South Wales. So Joel, thanks very much indeed for making the really long trip to be with us.
Thank you for the invitation, and it's good to be here. It's about 2 a.m. I think in Sydney, so I'll struggle with the jet lag. So I thought I'd just appropriate being here and talk about this before I get into the science, before I get into Future Minds, just to show a couple of my artworks from many, many years ago. I don't have much time to do painting and drawing, but I went to art school and studied painting, drawing, and film as well. And it was really the discovery process that fascinated me doing art and also in doing science that I really fell in love with. And in the end, up until now at least, science won, and the discovery thing about science really would grab me, and I became addicted to discovery. So, as Adam mentioned, this thing called this Future Minds Lab. I'll just get a very, very brief introduction to what that is. You call that something like a full stack lab, but that's another way of saying we do everything from the fundamental discovery. So we just study everything from mental imagery, memory, consciousness, intuition, decision making, and a few other things as well. Some clinical work. We turn that knowledge, that discovery into papers, talks, books, workshops, things like that. And then part of this new venture is to turn that knowledge into services and products. So we do that with our own spin-outs, our own companies. We work with corporates and startups, just trying to turn everything we know about psychology, the mind and the brain, into things that can make a difference for people. So that's what we're doing now. We're a team of about 20 people in Sydney, Australia. This is a couple of pics. But let's move on to why we're here. We'll talk about aphantasia and imagery. So as Adam touched on, a lot of what we have been doing in the lab for the last 10 years is trying to find more objective, more scientific ways of measuring things in the mind. And we've done that with imagery, we've done that with a few other things, with hallucinations, with intuition. So moving beyond interviews, beyond questionnaires, although those things are great, trying to get a more direct or a more objective way to measure things in the mind. So a super brief history of measuring imagery, as Adam discussed, questionnaires, the VVIQ is the gold standard, the classic. Then you can move on, go into literature, reaction time, so getting people to imagine things and then showing them something and see how quickly or slowly they react to it. Mental rotation, that's something you may have heard a little bit about. So typically rotating, this task where you've got to rotate something in your mind. So the spatial 3D rotation, or you compare two objects, are they the same or are they different? And one's a rotation, is one a rotated version of the other one? Turns out that's kind of a little bit different to imagining an object, imagining a color red, but it's more about three-dimensional spatial capabilities. Other ways people have measured imagery is looking at its effects on perception, right? So you imagine the color red while you're looking at the color red and you just measure how those things interact. You need to be very careful when you're doing that because we know that when if you do those things simultaneously, you're paying attention differentially to that red. And so you're measuring the effect of attention and imagery. So in the work we've been doing, we've figured out a way to separate out the effects of attention and imagery. So at this point, let me say, if anyone has any questions, I'm happy to stop and have, because we've jumped into a bit of data, some data slides, and so if you have any questions for clarification, Just throw up your hand and hopefully I'll see you. So what we do is we tend to separate imagery. So let's say you're going to imagine an apple like this. Then we get people to stop imagining the apple. Then we measure the effect of that apple, the imagery of that apple, on a perceptual thing, on perception. So the imagery and perception are separated in time, and that lets us separate out effects of attention. So we can use a perceptual stimulus to measure the strength, if you like, of mental imagery. So how do we actually do that? We use a class of visual stimuli or illusions called bistable ambiguous stimuli. So you guys have probably seen things like this, the vase in the middle there, or two faces, or this wireframe cube they call a Necker cube. So there's different ways of interpreting these. Either you see the vase or you see the faces. And if you're looking at it, you see your perception, your awareness will alternate back and forward. These are fascinating sort of illusions themselves. And perhaps the most interesting, the most vivid version of this is something called binocular rivalry. And it's harder to demo this without giving you 3D red and green glasses. But if you do something like just put up a hand in front of one of your eyes, just block one of your eyes, so one eye is looking at me, and the other eye is looking at your hand, what's going to happen is you're putting your brain to a state of rivalry. Okay, your brain is trying to fuse an image of your palm and me standing down here. And if you could equate those in terms of brightness and strength, you'd actually get this thing called binocular rivalry. You'd see me, then you see your hand, and you see me again, and it would alternate back and forward indefinitely. It's very hard in practice. You probably can't get that right now because the brightness and the luminance and the contrast and things like that are not equal. But is there a question up there? Yeah.
Yes. Is this the same with people who alternate and don't have stereopsies?
Not 100% the same, but it'll still work. So this is a sort of form of binocular rivalry. In the lab, when we're doing it, we use very different patterns, normally a red and green pattern, and we can control that with little 3D glasses or with mirrors, and there's different ways of doing that. And so they're quite easy to equalize the strength between those. And so this is a little cartoon of me. That's actually a brain scan of me, my brain. And that's what you might see. You see this awareness of the two patterns going back and forward. Again, this is when you look at binocular rivalry continuously. Now, when we want to use binocular rivalry, this illusion to measure visual imagery, we don't leave it on the screen. We don't look at it continuously like that. We flash it on briefly. So this is how this would work. So if we're going to use a red and green pattern like that cartoon I just showed, we will ask someone to imagine one of those patterns. In this case, there's a little faint green pattern up there. So we'll ask them to imagine the green. Then they need to stop imagining, and there's different ways of getting people to stop. Then at some point later, we'll just flash on that binocular rivalry, just on and off. So it's for a second or less. So it's not staying on, there's not this continuous vacillation, this oscillation, it's just on and off. And then people report which pattern did you see, the red or the green. So what we're looking at to see is how the content of imagery back here in time is affecting the way people see this binocular rivalry illusion. And what we find is that if people imagine a green pattern like this, and we flash on binocular rivalry, they tend to see the green. If they imagine the red, then we flash on binocular rivalry, they tend to see the red. In other words, the content of what people are imagining biases this binocular rivalry illusion.
Are you distinguishing between visualization and imagining? Because I can imagine green, but I can't see it.
Right now, I'm not. No, I'm just using I'm not being technical with those terms. I'm just saying visualizing visual content and imagery. And then the percentage of times or the number of trials in which what people imagine affects this illusion is a measure we use, an objective measure of visual imagery. We can go into more detail about that. So that basic sort of thing we discovered about 10 years ago now, and over many different experiments and different ways, we've shown that it's all kinds of details about it. It's a real effect. We use all kinds of catch trials and tricky things. We can map out how depictive imagery is and do things that sort of speak to this imagery debate that Adam mentioned between sort of Kozlin and Pilishin. But a nice way to sum up the data from these kinds of experience is that if you have imagery, if you have good visual imagery, you can think about it functioning like a weak form of vision, a weak form of perception. So it behaves functionally like that. So the ways we've mapped it out experimentally, the way it's represented in cortex and things like that, it tends to functionally behave like a weak version of visual perception. So that tells us that when you imagine something, it can literally change the way you see the world if you have imagery, particularly if you have very strong imagery. And this sort of binocular rivalry illusion is a way to hack into that and pick up on that and use that as a way to measure imagery. But it turns out it's a nice method to replace questionnaires, to replace interviews. It takes a little bit more effort. You need to get someone at the moment into the lab. You need to sort of set them up. It takes a little bit longer than simply filling out a questionnaire, but it is a bit more reliable. It lets us do a lot more things. It lets us map out how strong imagery is in different visual imagery in different parts of visual space. Look at all kinds of representations of colors and different shapes and orientations and motion and things like that. So the way I will talk about this often is that it's a diagnostic, or it's a sort of microscope to measure the mind. So using illusions like this is almost like a technology, if you like, to measure the mind. The same way you might use a microscope to look at bacteria, or you might take a blood test. We can build these tools using visual illusions or any kind of illusion to measure dimensions or aspects of the mind. So one of the things we did sort of a few years ago is to take people with aphantasia and use this technique to measure if they can get any of this bias, this priming effect. So the question is, how strong would their visual imagery be, even though they say they don't have it? So one possibility would be that imagery is still there somehow in the brain. It's still hiding there in visual cortex, and people just don't have sort of conscious access to it. Or it's a reporting issue. Or they're blind to it, but it's still there. So it's still there. It should still show up in this using this binocular rivalry illusion method. So when we tested a group of people with aphantasia, this is people with aphantasia here, no luminance, and in the general population, we see quite a difference in this priming, this bias effect. So it does seem that the feeling of not having a visual image is also related to this sensory representation in visual cortex, or this lack of this bias, or this lack of visual priming that we measure with this illusion. And there's different ways of looking at this data in terms of comparing it to a general population. I think this slide's a little bit better. But the people with aphantasia we measured here are down here around chance. So the way we do these experiments, red and green, there's two different colors, there's two different patterns in this rivalry illusion, so 50% is chance. And so in general, people who, I think we found one or two people with aphantasia who were getting some priming, but pretty much everyone else we've measured are not getting any priming. Any questions on? So it seems that a visual representation, most probably in visual cortex, is really missing in aphantasia. As I said, it's not a matter of metacognition. Yeah?
One thing, in talking to people that have it, we all seem to have a slightly unique version of it. How do you take that into account for this? Because if we're all having a slightly different experience, how does that then, are you assuming it's a baseline, everyone's almost the same, or have you taken the variation into?
Absolutely. So this was just the first step in sort of measuring, is there, so if we did find a measure of sensory strength of visual imagery, even though people didn't have any conscious access to that imagery, there'd be quite a different outcome. But absolutely. So in our population, in our data, just like in Adams, we find people that have only aphantasia with vision and all the other senses are fine, or people across the board have no. So there's all kinds of variations there, and there's degrees. What I think we don't know, I'll come back to the end, is the best way to think about it is it's on the spectrum. There's one spectrum of hyper to aphan, or is that down here categorically different somehow? Or what is the best way to think about that? So,
I mean, just coming back on that, I mean, it seems to me that people with, taking the vision element people have different abilities with vision, but you know, I couldn't even begin to understand why they vary, but they do seem to vary between people I talk to. I wouldn't even know how you go about measuring that variation.
In terms of the perception, visual, yeah. So we can measure that. So you do see, you see individual differences in pretty much everything you can measure, although compared to most things, the differences in imagery are much larger than most sort of forms of visual perception.
Okay.
Yeah. Right, so we do a lot of imagery research, and so I'm happy to talk about, I've sort of taken a sample of a subsection, some new experiments we're doing now which speak to aphantasia, but we're doing a whole lot of other things in different areas to do with imagery in the general population as well, and some clinical things as well. So I want to start off just by talking about visual attention or selective attention. So Adam mentioned this idea of this top-down voluntary control, and we use the frontal cortex to do things where we just put our minds to it and control. So attention is one of those things, and there's two sort of main types of visual attention. There's one which is called exogenous. That's if I clap or it's a loud noise, it's going to grab your attention. So things in the environment will grab your attention whether you like it or not. Then there's another type, which is kind of more like imagery, where you voluntarily pay attention. So while you're listening to me, you can decide I'm boring and pay attention to the door or the clock or something else, or vice versa. And there's different subcategories of that, and there's a whole sort of literature on this. So they're the main two forms of attention. So we also have been doing some research on comparing imagery and attention in the general population. So here we wanted to sort of take people with aphantasia and look at different types of attention. And we've done this a number of ways, but I just wanted to show some data we have using this task, and let me just take you through the way this works. So this is what people are going to see on the screen, and it's running through time here. And so in that red circle, that little square, when that flashes like that in a sort of bolded way, that's the exogenous cue. That grabs people's attention. They're looking at the cross in the middle, and then a moment later that star comes up and they have to respond to that star. And so that would be a valid cue because the square is grabbing the attention and pulling it off to the right side. And over here you'd see it's pulling off to the left and the star's on the other side. So that's an invalid cue. So the idea is you can measure reaction times here, and when the cue is invalid, you're going to be slower. And that's a classic. There's literally thousands of papers using these kinds of paradigms to study attention. Then you can do a sort of endogenous, where you have an arrow there and you have to tell people to move your attention over there yourself using your voluntary own steam. The paradigm is kind of similar. Instead of that flashing thing, the arrow's there and you have to move your attention to one side or the other. Again, you can have these valid or invalid types of trials. So we can look at data like this. So I've separated this, looking at reaction time here, people with aphantasia and people with imagery. And in this data here, we would also, I'm classifying sort of control, or people with imagery, people without imagery, both in terms of VVIQ and also the binocular rivalry illusion as well. So we use both those methods typically to find people with or without imagery. So this is the exogenous cue, that flashing square that's grabbing people's attention. And you'll see that the slower, the higher bars there are the invalid. So people are slower when their attention's pulled off to the other side, and they're faster when it's on the same side. And you can see there's a bit of difference there, both people with imagery, people without imagery. Now we look at the endogenous cues. You can see people without imagery really don't get, there's a little bit of an effect there, but really don't get much of an effect, or much of an advantage or disadvantage of having the invalid cue compared to people with imagery. So this is interesting because it starts to tell us that people without imagery may also have some differences with endogenous attention, but not this exogenous attention, which kind of makes sense because they both, as I said, involve some kind of voluntary effort to apply attention, just like applying imagery. Yeah.
I was just wondering if the difference was still significant in the
In this one, no. So I've taken the stats out of this, but no, it's not. But it is here. Yeah.
Why are the aphantasia reactions slower than on both? On endogenous? I'm not sure if I'm reading it right. It's a bit confusing, but
In general, or here?
like on the right hand side,
Yeah, it's really that. So it's the invalid cues are fairly similar. I think it's the valid cues the difference. So it's this bar here if you compare to that bar there. So yeah, it's not the overall slower. I think it's just they're not getting advantage of when attention is in the same location as the star or the task.
They look a bit slower overall on the left-hand graph, but is that not...
They did a little bit, yeah.
Yeah.
So as I said, I think this sort of speaks to some kind of overall. This is a new project where we're measuring attention in a couple of other different ways. So we didn't want to rely on just that one kind of task, even though that's the classic attention tasks. So we've got a few other ways and ways of slicing up attention there. And there does seem to be some subtle differences, particularly with this voluntary endogenous attention. And that makes sense because people for a long time have talked about this relationship between imagery and attention. So next I want to show you some data. We've been doing a lot of things for a number of years on working memory and visual working memory. So Adam mentioned working memory. So visual working memory is a sort of subcategory of this general working memory category. So when you're holding information over just sort of short periods of time, and in particular when we're talking about visual information, the kind of task we do is something like this, where you show people an array of different, we're using little striker patterns, different orientations. And you might just show them 1 or 2 or 3 or 4 on different trials. And all people have to do is try and hold those little oriented striker patterns in memory for typically 5 or 6 seconds, and then we'll randomly pick one of those which will flash up, and that probes have to report if that's a different or different orientation. And the kind of data this is... What happened then? Even with one? Interesting. Let me just show you this next one. So this is data. This is a classic data. You see this set size effect. So when there's 1 or 2, people are really good at this task. So this is people with imagery I'm showing you right now. And as you get more and more classic, there's a lot of data showing this. You gets harder and harder, this capacity limit, this bottleneck, and you get worse and worse. So this is standard kind of thing. So what's it going to look like? People without imagery, people with aphantasia, and we see the data looks pretty much identical. So again, this is people defined without imagery, again with the VVIQ, the questionnaire, and with the binocular Rivalry illusion both.
Tried it for 107 seconds.
Sorry?
We tried it for 107 seconds. Because I've had
We haven't with this.
7 seconds is the limit on short-term memory.
It is, yeah. So there's different kinds. There's very brief memory, so less than a second, and then there's iconic memory, visual persistent kind of memory, and then you get to the longer terms of memory. Here we're just sort of looking at the kinds of working memory in that zone where other evidence suggests that people who have imagery use it as a mnemonic tool. Yeah.
I've got to ask why, because
Why?
I'm
Sorry.
surprised at the result.
Well, yes, I will surprise. Yeah. So I don't have time to go into all in this data in a lot of detail, but it turns out there are differences. You need to just probe them. You make things a little bit more difficult, or you start. Let me show you the next slide. So one thing we started doing is if you ask people without imagery, so how are you doing the task? They tell you quite different answers to someone with imagery. So for example, when I do this task, I have imagery, so I pull them out of the pattern here, like this, I go, oh, I hold them in imagery, wait 5 seconds, and then the thing comes up, and it's like, oh, there's imagery. Someone without imagery won't be doing that. So when you give the people sort of questions like this, to remember these visual patterns, I tried to imagine them. So people defined as aphantasic will report that still not 0, but much less than people with imagery. And then you can do things like, I haven't got the data here, but you can flash on bright background. So you're on the computer screen, you flash on a bright luminous, bright background, and if people are using imagery, their performance goes down. If they're not using imagery, their performance is unchanged. So you can start to use these sort of disruptive techniques to probe how people are performing these memory tasks. So Oh, that's another one. So this is a different sort of an inverse question. So this is the same subjects, the same participants in the data I showed in the last two slides. So here people rehearsing these different angles. So saying, 45 degree, 45 degree. And again, people define the aphantasics report using that kind of propositional geometric strategy much more.
Sorry, is there almost a grading system that you're using a generalization that one size fits all? And if you appreciate that it's very wide spectrum, like hearing or visual impairment or whatever, so then would that not make a complication where somebody would be better at that, but there's a certain percentage that really won't be able to do it? Like, how can you get a generalization at the end?
Depends on, I don't mean to make a generalization, so I'm just, so the way I'll be talking is more, we're on a quest to figure out the mechanisms in the brain of imagery. What creates imagery? Why do some people not have imagery? What's the difference in the brain, and how can we best understand that and model that? So that's where I'm coming from. So I don't mean to generalize, or I might accidentally use language that suggests I am, but I don't mean to do that. No. So it turns out, so if you just look at people's performance in this sort of working memory task, with or without imagery, they look very similar. But when you start probing them, and there's other things when you go into detail, people with imagery are better at the task around vertical and horizontal. People without imagery, they don't show that bias. And there's other things like that which start to suggest there are differences with these visual patterns. But again, if you don't look at, feel like the strategy people are using, you just look at performance of memory, you may not realize all these underlying differences. And that's kind of one of the problems with the visual working memory, the working memory field in general, is that they often don't ask about strategy. They assume, and even across the normal population, or if you take out people with aphantasia, you see a difference in strategy. And you actually ask people to try and use a different strategy. Try and use imagery, try not to use imagery, and the data will look different, and the brain areas also look different. So you can get similar performance using different strategies, but when you start probing the mechanisms, things look quite different. So I wanted to move on to some stuff that Adam mentioned something interesting earlier, that he's got reports, as have we. Can we come back to the question at the end? Thanks. That people without imagery, and you guys can tell me if this is true, don't always enjoy reading novels as much as people with imagery. There's this sort of reports and discussions about that online. So there's murmurings there. We can come back to that. So we did this experiment. So memories, episodic memories about the past, ideas of what you might do in the future, scary scenarios typically might involve some imagery if you have imagery. And also in the clinical literature, which Emily Holmes will talk about in more detail, there's strong theories that imagery holds a special relationship with emotions. And this taps into anxiety disorders, PTSD, and things like that. That if you imagine something scary, it will fire up your emotions more than the semantic or the propositional ideas about that thing without the images. There's some data to support this in terms of self-report questionnaires, but we want to sort of try and tap in and measure this a bit more. So we used skin conductance, right? So that's a measure of, sort of a proxy measure of how aroused or how scared people are. So you put sort of measuring how much people sweat. And then we devised this paradigm where people are sitting down in front of a computer and we have these sort of little vignettes, these stories, and they're quite scary stories. So they're sort of sitting in a dark room, we're measuring the skin conductance, and you're in the water, the sun, there's a flash, something moves, there's a shadow moves underneath you, you turn around to the beach and people are screaming and pointing at you, and it keeps going like this until you see a fin and then you see a mouth, and then it's a shark attack, right? So you're living in Sydney and there's beaches, that's a thing. But there's also a rock climbing version, there's different versions of these we have, which all kind of lead to something not very nice. And so the idea is there's no pictures or anything, you're just reading these words, we're measuring your skin conductance. And so typically, again, this is what I'm going to call control, so people with imagery. This is what you might expect, this is what you kind of typically expect with emotionally arousing stimuli. That skin conductance measure goes up over time, so on this horizontal axis we have the time during that story's on the screen. So it keeps going up, and people, if you ask them, they sort of feel more and more sort of emotionally aroused or scared. So the question is, what's going to happen, what's the data going to look like for people without any imagery. We see a difference there. It's not flatlining, but it's quite different. It's significantly different from people with imagery, which is interesting. So it's a proxy measure. So I'm showing you a physiological proxy, a measure of something. I'm not looking at the amygdala, I'm not looking at brain areas, but it is suggestive of less emotional fear response to these stories we're showing. And that's just showing the average of the two. So just before I answer that question, so we wanted to make sure, we wanted to sort of go a bit further and think, well, is this something about imagery or is it something more general about emotions? So we did a version, if you like, a perceptual version. So we're now going to do exactly the same thing, but we're going to show people scary images, or we're going to show them baseline not scary images. So there's no story, there's probably not too much need for imagery here. So we're going to show people a slideshow of different scary images, and we're going to do the same thing. We're going to measure their skin conductance over that same time period. And when we do that, we see the two groups, again, with or without imagery, look pretty much the same. There's no difference really in terms of stats between those two. So when the stuff that is there, the images are there on the screen, things look very similar. When there's just words on the screen, the difference with or without imagery seems to change the physiological response. Right, so this, I think, supports this idea that comes up a lot in clinical psychology and psychiatry, that images do hold a special relationship with emotions. And so if you do have imagery and you're thinking about and imagining your plane crashing over and over again, it's going to drive your emotions in a way that thinking about a plane crashing propositionally without those images is just not going to drive emotions in the same way.
Has there been any indication that people with PTSD have better visualization or less?
Yeah, so there's data showing, so one of the sort of defining characteristics of PTSD is typically these involuntary traumatic images that spontaneously pop into mind. But there's other studies showing that in general, non-traumatic imagery is also more vivid as well in people with PTSD. Yeah.
Did you ever use real accounts of sort of horrific events instead of stories? Like, instead of like a fictional event where you're struck attack?
Yeah, I haven't done that yet. One of my students is doing that at the moment in terms of episodic memories and trying to sort of delve down that literature. But yeah. Yeah.
I don't think when I'm recording events, I don't think I store emotions. I think emotions come in real time. I don't think I store. Have you looked into whether that kind of event you're just talking about, whether we can access stored emotions? I know it's not a sense and true sense of the explanation of the word, but if you looked at that side of it, whether we can actually recall emotions as well.
We haven't done that yet, no, so I can't speak too much about that. Yeah, it would be hard to tease apart.
I don't know how you know the difference between the two, really. But
yeah, that's interesting. Yeah.
I have a question related to the working memory capacity task. If you put the disruptors to a verbal disruptor instead of image, did it affect aphantasia more than control?
We're doing an experiment now, but the logic is that yes, it would be the inverse relationship. So you have people doing some kind of saying words or counting or doing things like that, and should disrupt people that are not using imagery in these tasks.
Do you see an interesting question about if advertising and propaganda could influence authentic less, or are you not looking at that question?
It's an interesting question. Certainly, if it.
Because I wonder that about.
I think there might be a difference, particularly if emotions come into it. Again, if you look at the data I just showed, then in certain scenarios where it might be on the radio, or there wouldn't be, if you're watching it on the television or something, and you're seeing the thing, it may not be too different.
Mm.
But yeah, if you're relying on imagery there to build up some kind of association or some kind of learning thing, then I think there would be differences. Yeah.
The data you're talking about is all the emotion, I suppose, is all data attached to frightening and scary kind of emotions. So have you looked at other kind of, other, it's a whole spectrum of emotions?
Not yet. No, we started here because there's this big sort of nasty emotions are always much stronger than nice emotions, like in terms of brain response and physiology. So if you want to start somewhere, unfortunately, you start with the negative, they show bigger effects. So we start there, and then we start to look at real memories and future thinking and episodic memory and this kind of thing. Yeah. So not yet.
Because I'm sort of, as I was listening to it, I was wondering around, like, there's something about logic, isn't there, as well, that when you're looking at scary images. So I think my memory attaches to strong emotions. And I feel emotion when I listen to music and when I read something. But I don't actually feel scared very often by something I've read or seen even. I wouldn't be scared of a picture of a snake either because it's not really there.
Wait, when you remember, do you have imagery or not?
Sorry, no.
So if you remember a time when you were scared, do you feel scared again?
No, I don't.
But you knew you were scared at the time?
Yes,
yes.
yeah.
Okay.
But I can feel, but if I remember a time when I was sad, I can feel sad again. And when I remember a time when I was happy, or if I think about my children, then I can feel love, or I can feel joy. But when I think about the birth of my children, I can remember aspects of that, although I can't visualize it.
Yeah.
But the reason I'm wandering around that scary emotion is showing people pictures of things that are not, or like talking about a narrative based around Jaws. It's kind of unrealistic in terms of their frame of life reference.
Yeah. So it was, as I said, it was a starting point. It wasn't meant to be representative of all the full spectrum of emotions in everyday life. Absolutely not. So that's as we expand and do more and more studies on this. Yeah. We'll just do one more, then we'll keep going. We'll come back at the end. Yeah.
Do you have any intuition on how these results might, or rather, do you have any intuition on how these results might relate to non-visual imagination, like auditory, like other senses?
So, yeah. So I presume that there'd be something similar happening in all the senses. So typically, what happens is because we're visual creatures and like almost half our brain is dedicated to visual processing, visual cortex is huge. Often we start with visual imagery. Visual imagery gets a lot more attention than any other type of imagery typically. So often our approach is to start with visual imagery, see something, and then look at expanding it to other forms of imagery. Yeah. So I would generally predict, yeah, we generalize. Yeah. So let me just keep going a little bit, and we can come back to some more questions. So just quickly, the lighting may not be too good. How many of you can see red in the center there? Okay. And now? How many? So show me again. You see red, and do you guys mostly don't have imagery, I presume? Yeah. So that's interesting. So there's no red there. So as I cover these up, there's no red coming off the screen hitting your retina. So this is when I said before, we're using illusions as sort of techniques or technology to measure the mind. This is something called neon color spreading, and the particular arrangement of these circles is a way that sort of makes your brain feel and almost hallucinate red. So you're seeing red and there's no red there. There's no red hitting your eyes, your retina. So you guys are having, if you want to be generous, an imagery-like experience of the color red, right?
You mean a complete circle of red?
I mean red in here.
Right.
No? So no one sees red in there?
That's just on the white.
So you see no red in the center? All right, so I can see red. It's very subtle, but I see red. So I see, if I look in right in the center, I see a very subtle red there. So, see, we did a scientific experiment. Yes, a little glitch in the beginning there. We did some agile science there, and we corrected it. So this is an example of the kind of thing we're also doing, looking at what we're calling phantom perception or involuntary types of sensory experience. So where there is no information hitting your eyes, but people are experiencing. And so the question that someone else brought up, this idea of involuntary and voluntary imagery, that's something we're quite interested in terms of the spectrum of these kinds of illusions right through to PTSD, where people describe having involuntary imagery. So when we this is an online poll we did with, I can't remember how many people without imagery, and there's a few people with aphantasia reporting no red there, but reporting red there, but most don't see the red there, like you guys. So that's why I was kind of getting excited there. Okay, it's different. So now what we're doing, we can use that binocular rivalry illusion to measure this kind of experience. We have like a mini binocular rivalry display, and we can measure this phantom red, this phantom color experience in a similar, objective, reliable way. So that's a little teaser there of where we're going. So next I want to sort of jump course a little bit and talk about the brain mechanisms of imagery. And this is, as I said, this is research that's been going on for a number of years now in my lab. And so it's sort of looking at the whole spectrum of imagery strength and imagery vividness, not necessarily looking at people without imagery completely. But I think it still speaks to the theme of this conference. So the question is, what causes aphantasia? And we started this sort of quest, or this sort of journey by looking at what causes strong and weak imagery with a sort of simplistic bottom-up approach. So the first part of the cortex, which receives information, is primary visual cortex. It's a little pointy bit right back here. And we see huge individual differences in size of visual cortex, so 3 or 4-fold. So one person's visual cortex can be sort of many, many times larger than someone else's. And we see that relates to elements of visual perception. So we were wondering if that visual cortex size would also relate to dimensions of to visual imagery. So that's what I'm talking about. That's looking at one hemisphere of a brain, and that particular purple pointy bit there is that sort of what they call primary visual cortex, or visual area 1 is another nickname it gets. So there's 1, 2, 3, 4, there's multiple visual areas. So you might see it as primary visual cortex, or you might see it referred to in a slide as V1, just means visual area 1. So we have ways to map that visual area out. We functionally map it in an fMRI machine by showing people different kinds of patterns. So what I'm talking about the size or the area of that. So if you take the cortex and flatten it out, iron it out, and then measure that area, that's what I'm going to be talking about here. So we do that exactly. We measure people's imagery, and then we put them in the scanner and measure the anatomy, the functional anatomy of their visual cortex. So that's what I'm showing you here. So that's that V1. I said it's the area size of that little purple bit there. Comparing that to imagery strength, measured using, again, that binocular rivalry illusion I talked about earlier. And we see an interesting, significant correlation there. So people with stronger imagery or more priming actually tend to have a smaller visual cortex. Right? And it's actually opposite in visual perception. So otherwise, people who have more precise visual perception tend to have a larger visual cortex. So that's kind of when we first ran this experiment, that's what we were actually expecting to see the inverse relationship. So it's always kind of interesting when the data comes out the opposite way to predict it. And it's interesting because there's some data across different psychological and neurological situations that also suggest people with schizophrenia, PTSD, and Parkinson's disease tend to have elevated, very strong imagery. And there's some now emerging data that visual cortex might be smaller also. So that's, as I said, anatomy, brain anatomy. So next we wanted to sort of take a step further and start looking at functions. What are the functional causes of different types of imagery strength? I'm going to show you some data here. It's again using MRI, but functional MRI, to look at a resting baseline activity in the brain. So I'm going to be talking a little bit about excitability of the brain, resting state. So one thing we know about the brain is it's always active. So this idea that you only use 10% of your brain and things like that is not really realistic. So your neurons in your brain are firing all the time. They can't switch off. They can't be quiet. And so I'll be talking about this resting activity. So some people have quite some more quiet brains, or there's less noisy neurons just firing randomly. Other people have noisier brains. So that's what we're kind of measuring here. But everyone's brain the neurons are noisy. They just randomly fire all the time. So what I'm showing here is, again, imagery strength on the vertical and the horizontal. I'm showing you a measure of this resting activity from a functional MRI. And what we see is an interesting, stronger imagery equals a lower resting state, not higher. So people with a quieter visual cortex tend to have stronger imagery. So that's using MRI to measure that, and we can measure things in this excitability, this resting state, in a couple of different ways. So we can use something called transcranial magnetic stimulation, and that is, here we go. It's using a little sort of wand about this big, like a little sort of squash racket, and it actually is a sort of very strong electromagnet in there, about almost 2 Tesla. So it's like a big fMRI magnet pushed into a little thing. And when you put that over someone's head and switch it on just for just sort of 2 milliseconds, it induces this strong magnetic field, which induces activity in neurons in your brain, just for a split, less than a second. So when you do that, you see something. So this is a tool we use in research in all kinds of different ways. And the way I'm going to talk about it here is when people's brains are quieter, you need a lot more power in that TMS thing to create a visual phosphene or hallucination. So if I stimulate the visual cortex, I can make you see something, to make you hallucinate. And the noisier your brain is, the easier it is to do that. And the quieter your brain is, the more I have to crank up that power more and more. And so it's it's a tool to measure how noisy or how quiet your visual cortex is. Yeah.
Just ask, is that activating the bit that I would normally see when I'm dreaming, or is that going to be the bit when I'm seeing when I'm awake?
Probably both. So I'm talking about to see this thing I'm going to phosphate and make you have a visual hallucination with brain stimulation like this. I will stimulate the visual cortex. So again, I put it right back here on the
So
end.
it could be either that I would be seeing either an image I could see from a dream that could appear, or it could be something I could.
Yeah. So it's typically
involuntary though.
Yeah, it's typically what you see is like a little a flash of light, like in that little cartoon. So it's not. It's quite subtle.
Okay.
It's not like
Not going to be a physical thing.
you see a sunset. No, it's not like that. There is some data that if you stimulate other more object-selective areas, you can get more complex phosphines, as they're called, hallucination. But they're quite unstable and they're hard to get, and it depends on individual shapes of the brain and the curvature and things like that. But for now, we're just using this thing as a way to probe how quiet or excitable people's visual cortexes are.
Is it safe?
So the way we're talking about it here, it's a single click. So put it here up on the box.
Yeah.
If you're doing it over and over, so you may have heard, you can use this technique. It's an FDA-approved treatment for depression over the frontal cortex, and that's sort of rapid stimulation. So if you do a lot of these pulses over and over very, very quickly, you could sort of create these sort of waves of activity in the brain, which could give you a seizure. So that would be lots and lots of rapid pulses over some period of time. But we're not doing that here. We're using these single pulses, which is fine. Right, so what we're seeing here, again, using this quite different technique, that lower resting state, again, equals stronger imagery, or predicts the other. People, again, with quieter visual cortex are the ones with stronger imagery. So far I've been using I've been showing correlations, right? So stuff with the fMRI and using this TMS thing and correlating imagery strength to that. But we have other techniques, a different kind of brain stimulation technique called transcranial direct current stimulation. That's when you literally put a wire, say here or here, and run a very weak current through the brain, just running off a little tiny battery. And so you can use that technique to actually change the resting excitability of different brain areas. So the kind of thing we're seeing the correlation with, we can actually use this technique to change, up or down regulate, depending on the flow.